Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled Unraveling Platelet Phenotype and Function with Full Spectrum Flow Cytometry. My name is Lawrence Howes, and I'll be moderating today's presentation. I'm delighted to be joined by our guest speaker, Dr. Benjamin Spurgeon, the Associate Director of Diagnostic Hematology at Versiti. This webinar will discuss the advantages of full spectrum flow cytometry over standard tests of platelet phenotype and function, and how it can enable a sensitive and simultaneous detection of multiple platelet biomarkers. Following the presentation, we'll have time for a question and answer session. If you have any questions throughout the webinar, please feel free to submit them in the box to the right of your screen. So without further delay, I'd like to hand over to our speaker, Benjamin. I'd like to thank them again for presenting for us today.
Okay, so today's presentation is split into three parts. First, we'll provide some background on platelets and platelet phenotypes and functions. Next, we'll describe the optimization and development of a 16-color panel for analysis of platelet phenotypes and functions. And finally, we'll show some preliminary data that demonstrates successful application of the panel across different sample types. So platelets are small but highly abundant blood cells that circulate for 7 to 10 days, and platelets are made chiefly by megakaryocytes in the bone marrow, but also by megakaryocytes in the lungs, and they function primarily in hemostasis. Platelets express a range of specialized surface receptors which allow them to detect and respond to vascular damage. Platelets can aggregate with one another to form platelet plugs and prevent bleeding. They also express a repertoire of receptors that regulate immunity and inflammation, and expression of platelet surface receptors appears to be heterogeneous, leading us to believe that platelets circulate as distinct subsets with specialized functions and diverse capabilities. And a major challenge is to develop methods to identify these subsets, as such methods would be useful for a range of basic, translational, and clinical applications. Specifically, we think that platelet subsets could serve as sensitive and specific biomarkers for a range of pathologies. So flow cytometry is advantageous for platelet studies because it allows platelets to be studied in whole blood, which is the most physiological system. Ideally, we want to avoid isolating platelets from whole blood because it can introduce artifacts and affect platelet function and reactivity. Historically, assays use 2 to 3 core markers listed here to distinguish resting from activated platelets. And here the cartoon illustrates different mechanisms of platelet activation. So while platelets can be activated by soluble agonists such as thrombin, thromboxane, and ADP, a more classic mechanism of activation involves collagen and VWF, which are exposed after vascular damage. And specialized receptors stick to collagen and VWF, and downstream signaling results in activation responses such as granule secretion, and the contents of these granules can amplify platelet activation and help recruit additional platelets and even leukocytes to the site of damage. We also see activation of integrin alpha-2b beta-3, which mediates fibrinogen binding. And fibrinogen is a bridging molecule that allows platelets to aggregate with one another. Monoclonal antibody PAC1 recognizes the active form of alpha-2b beta-3 and therefore allows the detection of so-called pro-aggregatory platelets. After activation, adhesion molecule CD62P relocates from intracellular granules to the platelet surface, where it modulates interactions with leukocytes, and platelets expressing CD62P are said to have pro-inflammatory properties. Activated platelets also express phosphatidylserine, which can be detected using annexin V, and phosphatidylserine provides a scaffold for attachment of coagulation factors. Platelets that bind annexin V are said to have procoagulant properties. So while these 2 to 3 core markers have been useful in studies of platelet activation, in order to advance our understanding of platelet biology, it's important that we expand the size and scope of our panels, and spectral flow cytometry is an accessible platform that allows us to do this. So we designed a panel of 16 clinically and functionally important platelet markers shown in the table on the left. 9 are inducible markers that are expressed after activation, including annexin V, CD62P, and PAC1. There are also additional markers of granule and lysosome secretion, such as CD63, CD107A, and CD154, as well as markers of immune function, such as complement protein C3b and TLR9. 7 are constitutive markers with some exceptions. For example, CD36 can be absent in 3 to 11% of Asians and Africans. Other constitutive markers, such as GP6, can be internalized or shed from the platelet surface after activation, while CD42A and CD61 normally stable and abundant, which makes them ideal for platelet identification and gating. And we paired dim markers with bright dyes and vice versa, and tried to minimize spread into channels that need high resolution. And the complexity index for this panel was 6.46, which is reasonable for a panel of this size. So there is a simple no-wash workflow that requires whole blood collected in sodium citrate. Blood is incubated with a cocktail of platelet agonists and fluorescent reagents for 30 minutes, fixed and lysed for 10 minutes, and then acquired by spectral cytometry. So staining should be performed on fresh samples obtained within about 4 hours of blood collection, because platelet reactivity decreases over time. In addition, staining should be performed on unfixed samples, since fixation can result in artifacts and the loss of staining for some markers. So the figure on this slide compares basal and stimulated samples before, labeled pre, and after, labeled post fixation. So after fixation, we see a number of staining artifacts highlighted here, including increased levels of TLT1 and annexin V, while CD62P, PAC1, and others are markedly diminished after fixation. So successful phenotyping depends on the separation of antigen negative from antigen positive platelets. And this separation can be quantified using the separation index, which is useful in multicolor panels because it accounts for the spreading of dim populations. So antibodies were titrated using appropriate controls for inducible markers. Negative controls were unstimulated platelets, and positive controls were agonist stimulated platelets. And we wanted to obtain maximal resolution or saturation of these markers. For constitutive markers such as CD42A and CD61, we wanted to separate platelets from the background of coexisting cells, particles, and noise. And the optimal concentrations for all reagents in our panel are outlined by the red boxes, and pay attention to CD36 and GP6, since we'll focus on these markers on the next slide. So platelets are sensitive cells, and so reagents were titrated in the presence of CD62P, which is considered to be the gold standard marker of platelet activation. And we did this in order to find concentrations of antibodies that provide good staining without causing undesired platelet activation. So panel A, shown here, shows CD62P on the vertical axes and CD36 and GP6 on the horizontal axes. Inside the red boxes, we see that high concentrations of CD36 and GP6 cause an undesired increase in CD62P. However, we were able to find lower concentrations that provide separation of CD36 and GP6 without causing an undesired increase in CD62P. So in panel B, we checked if the combination of CD36 and GP6 caused activation or potentiated the response to agonist stimulation. The first 4 rows of this figure panel show CD62P expression on unstimulated or basal platelets. The control contains neither CD36 nor GP6. There is no change with CD36 alone, no change with GP6 alone, and no change with the combination of CD36 and GP6. The next 4 rows show CD62 expression on agonist stimulated platelets. There is no change with CD36 alone, GP6 alone, or the combination of CD36 and GP6. So we were happy moving forward with these particular reagents. So CD41 is sometimes included in platelet assays because it's abundant and useful for gating. However, we found that 2 of the most common CD41 clones, HIP8 and P2, block the binding of PAC1, as well as the binding of anti-human fibrinogen, FGN, which can be used as a surrogate for PAC1. So this figure shows PAC1 and anti-human fibrinogen on the vertical axes and clones HIP8 and P2 on the horizontal axes. And both clones show a concentration-dependent inhibition of PAC1 and fibrinogen binding, with HIP8 showing strong inhibition. And this is maybe not surprising because anti-CD41 targets the same receptor complex as PAC1 and anti-human fibrinogen. And we did not want to compromise on PAC1 staining because it's an important clinical and functional marker, so we dropped anti-CD41 from the panel. So calcium is essential for platelet function and for the function of proteases that regulate the expression of certain receptors. So using agonist stimulated platelets as an example, figure panel A here shows the staining of annexin V against GP6 in the topmost plots and against PAC1 in the bottommost plots, in the absence and in the presence of physiological calcium, which is about 2.5 millimolars. So without calcium, we're unable to capture a true representation of phenotypes, and we see just one population of platelets, which is annexin low and GP6 and PAC1 high. However, in the presence of calcium, we see a more accurate representation. Of course, we see annexin binding, which is known to be calcium dependent, but these annexin high platelets are low for GP6 and low for PAC1 because calcium-dependent proteases mediate the cleavage and shedding of GP6, and the intracellular cleavage and inactivation of the fibrinogen receptor so that it's unable to bind PAC1. And so platelets are partitioned into distinct subsets. We see procoagulant platelets, which are annexin high but low for the adhesion and aggregation markers, GP6 and PAC1. And we see pro-aggregatory platelets, which are annexin low but high for GP6 and PAC1. So it's clear that calcium is essential for accurate phenotyping, but the addition of calcium alone activates thrombin and causes undesired platelet activation. So in order to prevent this, we include in the assay buffer the thrombin inhibitor, PPAC. So figure panel B, shown on the right, illustrates the effects of calcium and PPAC on markers of platelet activation, CD62P in the top histogram and PAC1 in the bottom histogram. So in the absence of calcium and PPAC, CD62P and PAC1 are low, indicating an absence of platelet activation. Addition of PPAC alone does not change CD62P or PAC1. However, addition of calcium alone causes an increase in CD62P. Addition of calcium alone causes an increase in CD62P and PAC1. So calcium is causing platelet activation. But the addition of PPAC along with calcium is enough to prevent undesired platelet activation. So the panel also contains multiple brilliant violet polymer dyes, which can interact with one another and cause staining artifacts. And in order to prevent these artifacts, vendors often recommend the use of specialist stain buffers, such as Brilliant Stain Buffer, BSB. This buffer is widely used and doesn't seem to have any toxic or otherwise negative effects on cells, so we thought it would be okay to use on platelets. However, we later found that the buffer caused inhibition of annexin binding, C3b deposition, and PAC1 binding, all of which happen to be calcium and or magnesium dependent. And we later discovered that the buffer contains EDTA, which chelates calcium and magnesium. So we wanted to characterize the effects of Brilliant Stain Buffer in more detail. And from time course experiments shown in panel A, we knew that C3b and PAC1 peaked at about 10 to 15 minutes. So we thought it might be possible to perform a sequential addition of Brilliant Stain Buffer once C3b and PAC1 had peaked. But this wasn't possible, as shown in figure panel B on the right, where the blue histograms show unstimulated basal platelets and the red histograms show agonist stimulated platelets. In the first rows of the histograms minus BSB, samples do not contain Brilliant Stain Buffer. In the second rows plus BSB, samples contain the buffer from the start. And in the third rows plus BSB at 10 minutes, the buffer was added after 10 minutes. So for C3b, inclusion of buffer from the start caused severe inhibition of staining, but sequential addition did not affect staining. For PAC1, inclusion of buffer from the start caused a complete inhibition, and sequential addition caused a significant loss of staining. And the same was true for annexin, where Brilliant Stain Buffer caused partial or complete loss of staining. So when using new reagents or buffers, it's important to examine their effects on platelet function in order to avoid potential problems such as those demonstrated here. So while brilliant stain buffer wasn't compatible with certain markers in our panel, fortunately that was not a problem because there were no apparent staining artifacts among the polymers included in our panel. The figure here shows basal and agonist stimulated platelets stained with all 7 polymer dyes in the absence, minus BSB, and in the presence, plus BSB, of brilliant stain buffer. And the staining patterns are all very similar with and without buffer, except for C3b, which, as we showed on the previous slide, we already know is diminished by the buffer. So after performing the described optimizations, we wanted to verify the resolution and performance of the full 16 marker panel. So we compared marker resolution in double stained samples to fully stained samples under basal and stimulated conditions. The additional marker in the double stains was CD61, which was needed to identify platelets because some individual markers by themselves are too dim to allow platelet identification in the heterogeneous mixture of whole blood. And here we can see there is a very close overlap between the double and full stains outlined in red and blue, with minimal spread around dim and clinically relevant markers such as CD62P, CD63, CD107A, and others, where high resolution was important. In addition, panel B here shows that our hardest to optimize calcium and buffer dependent markers, annexin V, GP6, and PAC1, could be resolved as very distinct subsets. Panel C shown here is slightly different. This shows C3b only with basal blue and stimulated red samples overlaid for the double and full stains. We see no increase in C3b in the double stain, but we see an increase in the full stain. And this is because the complement C3b pathway requires the presence of a complement fixing antibody, such as an IgM antibody, and none of these antibodies were present in the double stain, but 2 such antibodies, CD36 and PAC1, were present in the full stain, and so this allows us to detect agonist stimulated changes in C3b deposition. Next, we tested the stability of basal and stimulated samples from 3 different blood donors after staining and fixation. So the graphs show the median marker intensities for samples acquired on the same day, the black icons, and after refrigeration in the dark for 24 hours, the red icons, and for 7 days, the blue icons. And markers were generally very stable for 24 hours, but some show a loss of staining at 7 days, especially the APC and P tandems, and therefore we recommend sample acquisition within 24 hours. We also tested interassay precision by running 3 separate assays on the same blood sample on the same day. So that's 3 separate reagent and cocktail preparations made by the same technician. And the CVs for median intensities were very good, ranging from 0.5 to 16%, with a mean of 5.1% for basal and 4.2% for stimulated samples. We then tested instrument precision by acquiring the same basal and stimulated samples 3 times, one after another. And the CVs were also very good at 3.9% for basal and a mean of 1.9% for stimulated samples. So to summarize the panel development strategy, we selected appropriate antibody dye combinations and minimized spreading. We titrated reagents and checked for artifacts related to fixation and undesired antibody interactions. We optimized buffers and added calcium and PPAC to provide accurate phenotyping and prevent undesired platelet activation. We compared double and full stains to confirm adequate resolution and rule out staining artifacts. And finally, we checked sample stability, precision, and reproducibility. So with a robust assay in hand, together with our collaborators at Boston Children's Hospital, Patricia and Martha, we used our assay to compare platelets obtained from cord blood to those obtained from adult peripheral blood. And our collaborators work in neonatal ICU, where preterm infants sometimes require blood transfusions, and this blood normally comes from adult donors. But as this figure shows, adult platelets and other blood cells, such as neutrophils, show increased function and reactivity, which results in a so-called developmental mismatch. And this poses a risk of thrombosis and inflammation for the neonates receiving transfusion. So with its high parameter capabilities, we hypothesized that our panel could characterize these developmental differences in more detail than existing assays. So we compared cord and adult platelets at baseline and after stimulation with ADP, the thrombin peptide TRAP6, and the combination of collagen and thrombin peptides, CRP plus TRAP6. Samples were analyzed using an algorithm called FAUST, which identifies and annotates cell subsets based on the markers that differentiate them most clearly. Here, FAUST was able to differentiate platelets based on high and low expression of 7 inducible markers, as well as the constitutive marker GP6. And the figure here shows vertical dashed red lines, which denote the high and low thresholds that were set on each of these markers. The platelet subsets identified by FAUST can be visualized on a U-map, where they appear as distinct islands colored by the phenotype, and the list of phenotypes identified are shown on the left side of this slide, numbered 1 to 21. The U-maps can also be colored by marker expression and used to verify the accuracy of labeling, where each island shows a relatively homogeneous expression of the markers that were used for annotation. So FAUST identified 21 subsets. The first subset, unlabeled, is a collection of residual platelets whose phenotypes did not occur frequently or consistently across all sample types and conditions. But the remaining subsets were all labeled, and they are ordered by marker positivity, with annexin positive procoagulant platelets in rows 2 to 12 and annexin negative platelets in rows 13 to 21. And the graphs on the right side of this slide show significant differences in platelet phenotypes across all conditions. And we'll summarize these differences on the next slide. To summarize, spectral cytometry allows the identification of novel platelet phenotypes and allows us to detect differences in subset frequencies between cord and adult blood samples. At baseline, we found that adult blood contained 13% more quiescent inactive platelets. ADP-stimulated adult blood contained 24% more activated platelets with a PAC1 positive pro-aggregatory phenotype that was consistent with secretion of granule contents due to CD62P and TLT1 positivity. CRP and TRAP-stimulated adult blood contained 20% more fully degranulated procoagulant platelets, while cord blood contained 16% more PAC1 positive proaggregatory platelets. TRAP-stimulated adult blood contained 34% more proaggregatory and pro-inflammatory platelets, while cord blood contained more non-aggregatory pro-inflammatory platelets. And this was an encouraging first application of the assay. And so far, the assay has performed very robustly across a range of different sample types, including thrombocytopenic samples, which have very low platelet counts. So I'd like to acknowledge the support from my former colleagues at Boston Children's, as well as the help of Sean Stowell's lab at the Brigham. I'd also like to acknowledge the team at Cytec for all of their fantastic support, and I'm happy to take any questions.
So we are going to wrap it up there. I'd like to thank our expert speaker for the informative presentation and discussion, and a big thank you to everyone for joining us online. We hope you found this a worthwhile session. If you have any further questions, please feel free to email me at editor@selectscience.net, and I will follow up with your questions for our speaker. Remember, you can download related resources in the tab to the left of your screen, and this includes the certificate of attendance. If you'd like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in just a few days. Goodbye, and thank you once again for joining us.